All right, so thanks everyone for coming today. As Justin said, we're going to be talking about the NIH data management and sharing policy, and we're going to approach this from a few different angles on a panel. So first off, we have Lisa Federer, who is the data science and open science librarian at the National Library of Medicine, and she's going to give us an introduction to what the policy actually says. Then we're going to have Milligan-McClellan, assistant professor of molecular and cell biology at University of Connecticut. And Kat is a real live researcher. We got our PhDs in the same lab. So usually most of the people that attend to talk to these calls are research professionals, but Kat's going to actually tell us a little bit about her research and then tell us about how this new policy is going to be impacting her work. And then I'll round it out with my perspective on how we as research support professionals can support researchers like Kat in complying with this policy. And even if you aren't directly data facing, I know a lot of you on this call probably are, but even if you're not, I'm hopefully going to provide some suggestions for how you can get engaged with the policy. So, and then after, we're each going to talk for 10 minutes or less, and then we'll just have a good Q&A discussion. All right. I guess, Lisa, do you want to share your slides?
Yes, thanks.
Welcome, Lisa.
Thank you. And thank you so much for the invitation. I'm really happy to be here with all of you today. So you should be able to see my slides. I'll move them in presenter mode. All good there?
Yes.
Okay, great. So like I said, thanks so much for the invitation. I'm really happy to be here with all of you. I'm going to try to keep my remarks relatively short, although there's a lot I could say about the policy, but leave time for Q&A. So as you may have heard, the new policy is covering really all of NIH funding and requires researchers to consider how they're going to manage and share their data. So this does include research funded in whole or in part by NIH that generates scientific data. So this includes extramural grants, this includes contracts, other extramural funding mechanisms that the NIH might have, like other transaction authorities. And it also includes NIH intramural research projects. So we do have about 1,700 principal investigators at the NIH who also will have to do this. And as I said, it really does apply to all of these types of funding that generate scientific data. Really, the only type of funding that is excluded from this policy is funding that does not generate data. So things like training grants, infrastructure development, where there's not really data being generated. In addition to people having not heard of this policy, I've also heard some people have some misconceptions about what is and is not included. And really, I think most people that get NIH funding should expect that this will apply to them. And by the way, there's a lot of information available online, and I've tried to give links throughout to make sure that you can follow up on any of this if you want to learn a little bit more. So the policy requires that grantees and other folks receiving NIH funding submit a data management and sharing plan. And I'll talk a little bit more about what goes into that plan, but essentially it outlines how the data, as well as accompanying metadata that goes along with that data, will be managed and shared, both during the active life of the project, as well as beyond. And also identify any potential restrictions or limitations on how that data can be shared or how that data can be used. So as we'll talk about, there certainly are things, particularly in the case of sensitive human data, where obviously it would not be appropriate to share all the data completely publicly. So these plans should identify where data cannot be shared for those sorts of legal or ethical reasons, and other sorts of restrictions on reuse. Importantly, this plan becomes part of the terms and conditions of the grant or contract or whatever the mechanism is. So once that plan is put into place, it is expected that the awardee will actually follow through that plan. There is a mechanism for making changes to the plan if necessary. Things change, of course, but it is expected that what is outlined in the plan, the awardee will actually do. So there are 6 major areas that should be included in the plan, which overall should be no more than 2 pages long. There will be a description of the data plus metadata and documentation that goes along with that. A description of any related tools, software code, whatever it is that someone might need to access, use, analyze that data. standards that will be used for the data or metadata. So this might be, actual metadata standards about the data, might be things like, file formats, those sorts of standards. Data preservation, again, during the active life of the project and beyond, as well as how and when the investigator will make access to the data available, and how long that data will be made available for. Fifthly, access, distribution, and reuse considerations. So licensing for the data, for example, whether there are restrictions for reuse of the data and so on. And then finally, who's going to be responsible for oversight of data management and sharing? Who's going to, ultimately the PI is responsible, but who within the research team will be responsible for sort of the day-to-day, making sure what happens in the plan actually, or what is described in the plan actually gets carried out. There's a lot more that I could go into here, but since we do have a brief time, I did give a link to this, the grant note or the guide notice that goes into a lot more detail about what's expected in each of these sections. I do want to talk a little bit about the description of the data and what is included in the sort of expectation for sharing and inclusion in the plan. So data that should be shared includes, as you might expect, of course, data that you would need to validate and replicate study findings. So if you have a publication that you're putting out in relation to the data generated by this grant, there should be adequate data shared to validate and replicate that. But in addition to that, there is also an expectation that data that results from the funding but doesn't necessarily result in a publication should also be shared. So that might include, for example, null findings that don't result in a publication, or things that maybe are not published by the end of the period of performance of the award, but any of that data that does result from that funding is expected to be shared. What is not expected to be shared is anything that's not really that sort of like final research data. So laboratory notebooks, lab notes, preliminary analysis, drafts of papers, emails to your colleagues, all of these sorts of things that are not really that final research data, not required to be shared. And also this policy does only apply to digital research objects. So things that are physical objects like biospecimens, not required to be shared. And there is also an exception in this for things that cannot be reasonably digitized. So if there are, paper collection things that for whatever reason cannot be reasonably digitized, that is a potential exception here. So I mentioned that there are, some expectations and limitations to things that are not necessarily able to be shared. So it's very important, of course, that when we're talking about human subjects, that the sharing happens within the constraints of informed consent. So participants in the study should be informed that their data is going to be shared. So of course, where there is, for example, a study in place that already has their participants consented and this wasn't included, that would not be permissible for sharing, and so that would be an exception here. There is, of course, considerations for privacy of the research participants. Certainly, in many cases, data can be de-identified adequately that you would not be able to re-identify a subject. But in cases where that's not possible, that there would be potential for harm or re-identification of the research subjects, that, of course, is a reasonable limitation. Also, where explicit law, regulation, or policy prohibits disclosure, HIPAA would be an example of this. And again, data sets that cannot practically be digitized with reasonable effort. So a general expectation that data should be shared, but an understanding that there are certain privacy, legal, ethical considerations that may limit sharing in some cases. So the way that all this works in terms of the sort of logistics of the grant process, the plan is submitted with the application for funding. This goes in the budget justification section. The peer reviewers may comment on the budget that is provided for carrying out the data management and sharing, but is not a scored aspect of their proposal. And the plans themselves are assessed by NIH program staff. And as I mentioned, these plans can be updated. So if you say that you'd like to share in a particular repository, but then you find another one's better or whatever, certainly specifics of the plan can be updated as needed. As I mentioned, these plans do become part of the terms and conditions of the grant, and program staff do monitor compliance with the plan at regular reporting intervals throughout the grant process. And compliance may factor into future funding decisions if PIs do not do what they say. There is the sort of mechanism for enforcement that is in place. Just a note that the data management and sharing policy is the minimum requirement for all of NIH, but there also may be more specific guidance that comes from particular NIH institutes, centers, offices, or even particular funding opportunity announcements. If you dig a little bit into the policy itself, you'll see that the guidance is very generic because of the fact that it covers all of NIH. We have 27 institutes and centers funding all different kinds of research. So it'd be really difficult to say all research should go into this repository or everybody has to use this standard. But there may be guidance like that that comes from more specific parts of NIH, so grantees should be aware of that for their particular funding opportunity announcement. And there are also other NIH policies that might apply. So for example, the genomic data sharing policy that applies to both human and non-human genomic data may also apply on top of what's required here. So I do want to share just a couple of resources for writing a data management and sharing plan that you may want to pass along to the researchers that you work with. One of the things that I think is really important to be aware of is that I think a lot of times people complain that they see this as an unfunded mandate, that they're going to have to do all this extra work and not get paid for it. But you can ask for allowable costs within your funding. This includes funding for the data curation and development of documentation. So sort of that during the research process, local data management while you're in the process of things, as well as preservation and sharing in repositories once the project is done. One limitation here is that any funding that you ask for does have to be spent during the period of performance. So if you have something where it's like, okay, you're going to put this in a repository, you have to pay yearly for every 10 years, that wouldn't be able to be covered under this because the period of performance would end before that period happens. Because of the fact that this is new to a lot of researchers, they may not know, like, what do I even ask? What does it cost to curate data? So the National Library of Medicine funded a study a couple of years ago by the National Academies that looked at forecasting the cost of data. And they give a really useful framework for thinking about, what are the things that I should consider when it comes to thinking about what I need to fund management of my data, both in the sort of active data collection period as well through ongoing preservation. When it comes to where to share data, again, I said that the policy doesn't outline any specific repositories that you have to put your data in. And generally, NIH encourages the use of subject-specific and open access data sharing repositories. And I'll say a little bit more about the link that's on the screen here. Where there is not a subject-specific or discipline-specific repository available, there are other options like PubMed Central can store data that's associated with a publication up to 2 gigabytes. There are a number of generalist repositories that are a useful place to put data that doesn't have sort of a natural subject home. And for larger scale projects, there is the STRIDES program that NIH has partnership with the major cloud providers that could be a useful solution for people that do have those very large scale, high priority data sets. I mentioned this link to the BMIC repository page. BMIC is the NIH Biomedical Informatics Coordinating Committee. And one of the activities of that group is to put together a website that has a list of repositories that are useful for NIH researchers, either in terms of where to put their data or where to find data that they might find useful. Currently, that webpage, which is linked here, is divided into 3 sections: fully open repositories, repositories that have some sort of limitation on who can submit or access the data, and generalist repositories that are, not subject specific. They'll take data regardless of what it is. This webpage, I will say, is undergoing a redesign and a revamp that I think will make it much more useful in the next few months. And eventually those will all be collapsed into one list that I think will be a lot more usable. But I think this is a great resource for, again, researchers that maybe have not had to do this before and are not totally sure where to put their data. Beyond the list of repositories that BMIC maintains, the policy also includes an appendix that is a set of desirable characteristics that also recently was issued by the Office of Science and Technology Policy with some additional information and guidance. But basically, these desirable characteristics outline 10 things that repositories ideally should meet. Things like they have persistent identifiers, they have plans for sustainability, they address security and privacy, these sorts of things. And there's an additional 9 characteristics for repositories that house human data. So just sort of a higher level of security and control, recognizing that we need to be ensuring privacy and security for human data. So lots more information at all the links that I've given, and I will share these slides with the organizers, and hopefully they can share this out so you have those links available to you. One sort of all-purpose link, if you forget all the other links, the sharing.nih.gov site is sort of a clearinghouse for information about this plan, as well as the other policies that relate to data sharing at the NIH. So that's a useful resource. They continue to add to that all the time, so definitely check that out. And I will stop there and look forward to answering any questions you have during the panel discussion. Thanks.
Thanks, Lisa. That was an excellent overview. Kat, are you ready to share your slides?
It would help if I wasn't muted. So there we go. Okay, let me find a screen that actually has. There we go. Do you see it?
Yeah, looks great.
Cool. Okay. That's my Anipak name and my Twitter handle. And thank you so much for giving me a chance to talk to you about the data that I use and the data that I generate. Most of my slides, most of my talks start with this particular title, Adapting an Evolutionary Model Organism for Host-Microbe Interactions, because that's what I did. And that's what my lab continues to do. I also start with a land acknowledgement. I'm not going to go into it. I only have 10 minutes, but I highly encourage folks to look at the landgrabct.org website, and that'll tell you where the land for the University of Connecticut came from and which tribes were affected by that land grab. Okay, so what does our lab do? Our lab is really interested in host microbe interactions, and we study the microbiota. And as people who work with large data sets, I'm sure, or data management, I'm sure many of you have probably had to deal with the data sets that we generate. In particular, we're interested in how the microbes in the gut, those complex communities of microbes, affect the development of the host, and what happens when we disrupt those microbiota. We know that that disrupts the development of the host. Can we repair that by adding back microbes or manipulating the microbiome in some way? Now, a lot of that work involves ecotoxicology, and I'm not going to go into great detail about that, but just to let you know that one of the ways that we study how the microbiota is disrupted and what happens, can we repair that, is studied in the context of ecotoxicology. Mostly what I'm going to focus on today is our work looking at how the host environment in history contribute to host microbe interactions. That's a really fancy way of saying that I'm interested in microbe and host microbe interactions in populations within a species. So we take one species that has lots of populations, just like humans have lots of populations, and we look at how those populations differ in the ways that they are affected by microbes or disruption of those microbes. Now, to do that, we use this animal 3-spine stickleback. It's a fish about 2 to 3 inches long, found throughout the Northern Hemisphere, and importantly, found in Alaska, which is where I am originally from. I'm from Kotzebue, Alaska, and it's also where I spent my first 5 years as an assistant professor. I was at the University of Alaska, Anchorage. The nice thing about doing this work in Alaska is that there are literally hundreds of populations of stickleback around the area that I used to work. So I was in Anchorage, which you can see in this inset is a city of about 290,000 people. That yellow line is a road system, and all the blue dots in there are lakes. And if we look at that in a different contrast, here we can see all of this blue. Each one of those is a body of water. Almost every single one of those has stickleback in it. And so we had access to hundreds of populations. These fish tend to come in 2 ecotypes, or they're studied in 2 ecotypes, freshwater fish that spend all their time in the freshwater, and anadromous that spend part of their time in the ocean. We're not going to go into great detail about that, except to say that that's the context in which I study these fish. Now, one cool thing about this, each body of water has its different microbial community, predator, prey, stress, things like that, that can affect the host microbe interactions between these fish and their microbes. And so our hypothesis is that these fish have evolved different relationships with their microbes. And so we look at fish from different populations. I'm going to focus today on 4 populations that we've studied quite a bit. Westchester Lagoon, which is a freshwater population and is in downtown Anchorage. That's the green dot. The red square is Big Lake. It's a large lake that has a lot of recreational use. Bear Paw Lake is relatively pristine and small, and then Rabbit Slough is an anadromous population. So that's 3 freshwater populations and 1 anadromous population. There's 2 big types of experiments that we do. The first one is just figuring out who's there. So we pop guts out of adult fish, and by popping, I mean we euthanize them using IACUC-approved protocols, and we carefully dissect, minimizing contamination, dissect the guts from euthanized fish. Then we isolate the DNA from those guts, sequence the 16S gene, which is unique to bacteria and not the host, although whatever. And then we do a bunch of bioinformatic analysis and come out with these pretty graphs. The dots that are closest together have microbial communities that are really similar to each other, indicating that their microbes are similar and they're in the same abundance or relative abundance. If the 2 dots are further apart, the microbial communities are less similar to each other. So our lab generates a lot of data like this. I'm just showing you the 4 populations that I highlighted in my earlier slide. But you can see here that we have at least 40 dots for each population, and that the populations have different microbial communities. That 40 dots means that we have files to go with each of those individual fish. And these are raw sequences. The data that is associated with this type of studies is, we have several different types of data that are associated with these studies. The first is raw sequences from the sequence data that we get back from, usually we use Illumina platforms. So we send off our data with primers to amplify that 16S gene. We get a bunch of raw data, and these are huge. We store them as FASTQ-GZ files. From Illumina, Illumina says these are typically 22 to 24 gigabytes, and we get 2 of these files for each run. We did all of these fish in 1 run. So for this particular experiment, we had 2 FASTQ-GZ files that were about 22 to 24 gigabytes when you unzip them. When we get that data, we trim it, we sort it, we put it into a .biome file, which is an awful lot like a CBS file. It's relatively small. And I just did a quick look through our files. Our .biome file for this particular experiment was about 600 KB. We have a metadata file that goes along with this. It's a .cbs file that's relatively small. We have a pipeline for all the bioinformatic steps that we did to generate these data sets. That's a text file. We have a README that describes the experiment, and it's pretty small. It's also a text file. We have one of these for every single one of these sequencing experiments. We have data output, analysis output, like graphs like this one, usually in a JPEG or a PDF or a, I think that's supposed to be ESV file. And then we have our images of fish. And the images of fish look like this. Each one is about 5 gigabytes. And for experiments like this, we have an image of every single fish. So every single one of those dots we have an image of those fish that we can do different types of analysis. So lots of different forms of data, some of them extremely big, others of them are relatively small. And all of this will eventually get onto a GitHub server. I haven't done that yet. Right now it's in the Google Drive, and I'm a bad person because of that. Okay, so the other type of data that we generate, I'm a microbiologist. We've isolated over 350 different microbes. Each one of these has a 16S sequence that goes along with it, and an image of the microbe on the plate, like these images here, and also a gram stain so you can see what that microbe looks like in high magnification. We have full genomes for about 20 of these microbes, and those will get published, and those will be submitted somewhere, GitHub. Probably, and then we have characterization of the microbes, usually in CBS files. And then the last type of experiment that we do is basic experimental setup includes making fish germ-free, so just killing all the microbes that are associated with them, adding back individual microbes or communities of microbes that where we've selected individual known microbes, or conventionalized microbial communities, which is just everything that fish is normally exposed to. And then we take images of every single fish and do growth and morphology analysis. We characterize the microbiota. We look at behavior, and I'll show you what that file looks like. And we do immune system analysis, which is either a QRT-PCR, which is relatively small data set, or whole transcriptome analysis, and which is similar to the 16S in size. And then we do experiments like this, where we make the fish germ-free, usually from a couple of different populations. We add back microbes to half of those are the CV fish, and then we add back individual microbes or an ecotoxicant like crude oil, perchlorate, antibiotics, whatever ecotoxicant we're looking at, and then we collect our fish. Now, I want to note that we do a lot of measurements at this stage, and so each one of those measurements is another CBS file or text file. And I also want to show this image. We have pictures of every single fish from these experiments. This particular experiment had 24 flasks, and each flask had 20 fish in it. So this 3 times whatever big number that is. And then we also have behavioral data with videos. There we go. 5-minute videos, and we follow the path of every single one of these fish. As you can imagine, these videos are pretty big. They're about 600 KB for each one, but we have a 5-minute video of every single flask that we've recorded, and then we have still images like this. And then we can plot out how different microbial manipulation affects the development of behavior of these fish. Okay, so how will this policy affect me? Luckily, I'm already pretty pro open science, and I have a data sharing protocol already that I've written for NIH and that our lab follows, including throwing things on GitHub and protocols.io. But I do need to figure out where to store our data long-term, especially the raw data files. I also want to do a shout out to OpenScapes for open science training. My lab went through their training last semester, and they taught us, gave us more in-depth information on how to use GitHub and protocols.io and others. And all of both GitHub and protocols.io are open source platforms where you can store your data, store your pipeline, store your README files that come with your data, etc. And that's it. I went 2 minutes over, I'm sorry. I have a lot of people in my lab. And the other thing I want to note is that we've gotten funding from a lot of different places, including NIH and Moore Foundation. Both NIH and Moore Foundation, and I believe NSF now, requires open source, or open access data to go along with the publications and the grants that we've gotten. So that's it. That's all I had to tell you about what we do and how this will affect us.
Thanks so much, Kat. That was awesome. We've got a lot of positive feedback in the chat about hearing about your research, and I am resisting the urge to launch into a data consultation with you right now. So.
Okay.
All right.
And using Google Drive doesn't make you a bad person.
Oh, I feel so guilty.
Probably could be figured out though.
Yeah.
Actually, can we, before we go on just for a second, can we dig into that just a little bit?
Sure.
Yeah.
Why, I just want to articulate why the guilt around Google Drive?
Well, yeah, go ahead.
Oh, for two reasons. One is that it's not easily accessible to everybody. You usually have to have a link that you share with folks, and it can be difficult to figure out what's open to everybody on our Google and what's not. So we can have a folder that is just like open access, come here, and we can have a link to that to our website, but it becomes difficult to figure out to organize things on that. The other thing is I know that storing your files, your raw data files as a zip file is not the best way to store your data, and we have data limitations on our Google Drive as well. And then the last thing is it's really nice to have a standardized information that goes with every data set that you have. So we have these huge sequence data sets, and we need to have information about primers and like all this stuff that goes with it. And it's much easier to normalize that on a platform where other people are submitting data, and we can see what data they're submitting with their sequences.
I can concur a bit on the Google Drive in that the lab I work in uses Google Drive for like while we're working on the project, but once we get around to publishing it, we usually upload to SRA.
SRA. Oh, awesome. Okay, thank you. Yeah, I'm definitely taking recommendations.
Yeah, SRA is basically, it's an NIH funded repository for genetic data sets.
Great.
So.
I'm writing notes. Thank you.
Tempted just to not present and let the conversation go from here, but I'll run through some stuff really quick and then we can have a better discussion. Does that sound good to everyone?
Thank you.
Awesome. All right, so I'm going to present about what I think we as researcher facing type people can do to help researchers like Kat with all this, with new data requirements. And we're lucky to have Kat because she's already thought a bit about data management and has some things in place, but we're definitely going to be encountering people who have, like Jana said earlier in the session, that have never heard of this policy. So there's a lot of ways we can help. All right, so the resistance that you might be facing is maybe not because you came to this call, but like you might think, well, I'm not data facing, like how can I help with this policy? But I bet even if you work on an HPC system on the hardware side of things, you've got data that's running through your system. So there's ways that you can interact and intervene just because you're talking to researchers. And all of you have probably seen some version of this data lifecycle where you plan your research, store during the project, do some analysis and visualization, and at the end of the project, it needs to go somewhere, either to get archived or to get published, depending on the type of data. And it's really tempting to assume that the NIH data policy only really affects the planning stage where you have to submit a plan with your grant, and also the sharing part, which is the part that is visible to the public. And it's what NIH is going to be able to see and evaluate to potentially make future funding decisions based on your policy. But the work that goes into data management actually happens throughout the entire life cycle. So whether you're working in an academic library and you have a data repository that somebody can put stuff into, or you're consulting on data management plans, or you're working in research IT and you're running storage systems or do data science consulting or things like that, there's places that you can help throughout this entire process. So just in general, I think the 4 things that you can do is to stay calm and help researchers also stay calm, because it's a big change, but you know, we can do it. It's a doable policy to comply with. Make sure you know who else on your campus is more data facing and be able to refer them appropriately. And then also bigger asks are to learn about data management best practices and then try to kind of sneak them into your own work. And then collaborate with your colleagues to solve bigger problems on campus regarding like the data, like thinking big picture about the data lifecycle and how what researchers have access to while they're working on their projects. So as far as staying calm, I have some stock objections. I think Lisa already talked about some of these and some stock answers that I give when people say these things to me. We already talked about the unfunded mandate. Some people will feel like it's just another hoop to jump through. I emphasize the selfish motives for planning ahead, because if you don't count how many gigs your data files are going to be and then multiply that by the number of data files you expect to have, you could end up in a situation where you don't have enough stable storage to store your data, and then you just start shoving it in several different Dropbox accounts and then can't find anything at the end of the project. So appealing to selfish motives is always a good idea if you have somebody who's resistant. And then also, like Lisa said, these plans are living documents. It's not written in stone what you said at the beginning. So as your project changes, you can change your plan. And there's also many acceptable reasons not to share data. So as far as referring to people, you don't need to be an expert in all of this, but it helps to know who the experts are to refer about specific things. So common resources that are on campuses that I've worked on are data librarians who will do consulting on these types of things. I also do consulting on data topics. I'm not in the library, I'm in a research computing group. But yeah, it's different on every campus. So just know where you can find people. It also helps to know what the data storage options are on your campus, because, depending on the type of data people have, like specifically thinking about things like HIPAA, there's usually only 1 or 2 spots that, if anywhere, that the data can go that are available to everyone on campus. So just, that's an easy thing to know and to refer people to. And also, like on a lot of campuses that I've been on, not everybody has access to the same resources, because there'll be like core facilities or support centers that are available to one college or one school and not to others. So just being able to be aware of those differences to refer people appropriately. But in addition to referring to people, you can also refer to resources. I'm not going to spend too much time on this slide because I think Lisa talked about most of this stuff in her part of the presentation. But I will point out that if you can send researchers to dmptool.org, anyone can log in and see. It basically turns the NIH requirements or any grant requirements for data management plans into a template that you can fill out. And depending on your university, there might be university specific guidance in there if you log in with your university email address. And then just thinking about Kat's problem with where do you store the data long term? I think people are starting to realize that there are appropriate kind of methods and times to think about using cloud resources for research data, and data archiving is one of them. And NIH has the STRIDES program that provides discounts in those areas. So bigger asks now. So you can learning about data management best practices is a good way to kind of sneak in these like broccoli type topics into more popular trainings. I've run in parallel a coding, like an R programming workshop series and a data management workshop series, same location, same time, things like that. And I consistently was selling out the coding workshops, and the data management workshops were attended. I got at least a few people at every one, but people are way more excited about going to learn Python or R than they are about learning about how to write a data management plan. So thinking about how you can integrate some of these topics into trainings that are a little bit more popular is a way to help. And also, as you could think about trainings for collaboration. So think about how to do a training with somebody like that is a data expert, and combining their expertise with yours to either make new trainings, kind of get a bigger picture of how the data landscape looks at your university, identify gaps, and then figure out ways to advocate to get those gaps filled. And this is stuff that isn't something that's going to, be done in a year or, even 5 years. Sometimes you just have to keep saying the same thing over and over again, and then eventually somebody who has control over money will think it's their idea and then they'll make it happen. But just being the voice there that advocates for these resources and keeps the conversation going is really kind of what's needed to make change. Yeah. So thanks for listening. I've set up a Jamboard so that we can kind of brainstorm about other things that we can do. Because like I said, this is just my perspective on how I see things, but I'm sure there's stuff that I'm missing. And feel free to argue with me if you think anything that I said is off base.
Tobin, can you maybe drop the Jamboard link into the chat? And I haven't used Jam before. Is that sort of like a Discord server? Oh,
sorry. It's basically like a whiteboard. So we can take notes, sticky notes, things like that. So just like a less structured than a Google Doc. If people want to draw pictures about how they feel about it.
Got it. Thanks.
Yep.
So we have a couple of questions that have come up, but first I just really want to thank our speakers. This has really been providing a helpful overview. And one of the things that Kat's presentation brought to mind was all of the different data that Kat collects. And this was kind of one of the questions was, of that enormous variety of data, what needs to be captured in the data management plan? And I guess that's sort of for Kat, but that's also sort of for Lisa.
The way I understand it, anything that we do analysis on, right? So our videos should be made available, the raw sequences, in case somebody wants to do their own analysis on the raw sequences, all the files that go with those raw sequences, the README files, the forgetting, not the CVS, the CSV files with all the data in it, the metadata that goes along with it, descriptions of how the samples were collected and all that needs to go and be readily available with people, as well as the primers that we used and the protocols that we used. The protocols need to go on protocols.io, which we're in the process of converting all of our protocols. And the images, raw images, I think eventually need to go onto websites as well. Although now that I'm thinking about it, we didn't do that with our last publication. So I need to make sure those get thrown onto GitHub as well.
Yeah, and I'll just add, like, as Kat was talking, I was like, oh, this is a perfect example of just like really understanding how complex the like repository ecosystem is. And again, going back to like, you know, people are like, why doesn't NIH just say put it in this repository? Because I mean, like, this is a perfect example. This is one study, and we're probably talking about, you know, a handful of repositories because different kinds of data, and you know, how do we make sure that we can tie all those things together? So it's definitely a bit of a challenge. And so yeah, I think it was a great example.
I had a follow-up question on that. So yes, once all the data is put in there, is the responsibility on the PI if someone wants access to figure out, mine that data, who's going to be the one doing that stuff?
So I'm pretty sure that it's, so you have to put a link in your publications as to where that data is stored. And so people should be able to access that. The GitHub, you can, if it goes into GitHub, you can make it public automatically. And so people can access your data that way publicly without having to go through you. If it's private, like I've not worked with human data, I work with Stickleback specifically, so I don't have to work with humans. But if you have human data that can't be made public, if I understand correctly, you can make actually, I'll just let Lisa answer that one.
Yeah, so as for human data, so there's a few different options. I think ideally we want to stay away from this situation of, you know, data available upon request. There have been lots of studies that look at journals that, you know, have data made available upon request. You, in practice, you know, can access very little of that data. So ideally, the data would go in some sort of a repository where it's there, and then the PI doesn't have to think about it, worry about requests, any of that. As far as human data, there are various different solutions for that. One is controlled access repositories. So there would be metadata that's publicly facing about what a data set contains. But to actually see that data, you have to put in a request saying what you're going to do with the data. It goes through a data access committee who makes sure that this is an appropriate use of the data, and only then do you get access to it. So that, I think, is a good solution for human data.
Yeah, the only reason I ask is I come from the imaging world, and then I'm thinking of one particular RO1 that I'm working on. There's like 4,000 patients. Each of them have 5 different imaging studies. They're like 10,000 images per patient. So like the sheer volume of like curating that data, making sure that it's available to public, storing that data, mining that data, even sharing it, I'm just like it's a little daunting to think about how much would go into making it shareable.
I totally hear you. That's a huge amount of data.
I will say that, like, so I know that for what I work on, which is targeted capture of human data, humans, because we're doing highly targeted capture of very few genes, it really isn't identifiable just from the data. Like, I know that
imaging data, you can actually, we also had to push through like defacing and all of that stuff, because you can literally make the person's face out of this data.
Yeah. That sounds like a different, that sounds like actually it may be a, you may be an exception to it, because you cannot share it without making them identifiable.
So I just had a follow-up question on that. So are there like specific resources like this designed for like imaging data? I know genomics has its own thing. Does imaging has its own set of rules or resources?
I don't know about any specific guidance on that, but there are repositories that are specific for imaging data. And so I wonder if some of them might have, guidance that might be useful for that. But yeah, I think absolutely in a situation like that's exactly an example of like the kind of thing that you can't de-identify it. So, that's something that you cannot just like put that up for everybody to see, So that would definitely fall under that, sort of exception.
So Lisa, we've had a couple of different questions around this non-compliance piece. So after the end of the funding period, non-compliance might impact future funding decisions. So I guess question one is, does this have an impact for the institution or for the PI, or both?
Yeah, both. So the policy does state that the institution is also responsible for this. So, I mean, obviously this has not gone into effect yet, so we have not seen examples of like how this plays out. My personal thought is that it would have to be a pretty egregious violation for the institution to actually like see consequences for the rest of their PIs. But again, we haven't, seen this play out yet, so I don't know exactly how that looks. But yes, the institution is also responsible for, compliance with the plan and the policy.
All right, and then another question we had is, does the NIH DMS policy mandate sharing all de-identified data that a lab might generate, or just the analysis?
So I think that depends on specifics of the type of data. Going back to, what the policy says is the data that you would need to replicate or validate the study. So that may not be like every single piece of raw data that like comes off your analysis machine. I think that looks different depending on what your study is, what type of data you're working with. So I hesitate to give like a blanket answer, but it would not just be like the final like analytical data. It would be like underlying data that again would allow someone to replicate that experiment.
Including code?
Yes, so the policy does include software, code, other tools. So, if you're writing your own sort of like in-house code, ideally that's going to be shared. If you're using a commercial analysis tool, you would ideally point to what that is. So yeah.
Okay, I have another question I want to feed you that we got in the document. Do people have workflows where funding requests and grant applications encumbered by such requirement need to be cleared by the research computing group to make sure appropriate funding requests for storage have been made? I guess that one's more to kind of the larger group. So actually, I'm not sure whose question that was, but if you want to elaborate on that a little bit, we have a minute.
I think that was me. I just was curious because obviously you run the risk of unfunded mandates if people don't request the funding in their grant proposals. So you know, is there mechanisms in place whereby, you know, any funding requests or grant proposals that go out gets run through a, you know, in our case, I think it would be our office sponsored projects or something where they review it first and say, oh no, you need to go back and look at your data management plan and put in appropriate funding for that.
So at University of Illinois at Chicago at UIC, we are in close touch with our VCR office, where our grants and contracts office and IRB offices are located, and we are trying to put together a mechanism in place. We are right now developing a cost model, and if someone checks in their IRB proposal that they are applying for grant and some of these requirements will be applicable, then we will have a mechanism to check for that line item. We don't have it yet, but we are moving towards it.
I think it might be self-limiting because I'm pretty sure that NIH themselves will probably kick back any requests, applications or whatever that don't have a fully developed and data management plan with costs in it. So they'll probably look at it very skeptically anyway. So if you haven't done due diligence on that in the first place, your chances of funding are low.
Thank you for raising this, Alistair. This is such a good point, and it really does speak to the role that we need to play to help our community. And I'm really sorry, but we're out of time. Thank you so much to our speakers today. So Lisa from NIH, thank you so much. And Kat, who talked to us about stickleback fish. And then Tobin Magle, who is helping us think about how research computing can play a role in here to help with this. The policy we didn't mention, I don't know if we mentioned this, kicks in January of 2023. And so this is definitely the right time to get ahead of it. Our next call we don't have a July call because I think we're all going to be at PERC. Please, if you haven't registered for PERC or if you don't know what it is, PERC 22, go ahead and if somebody could drop that in the chat, that'd be awesome. That's our annual gathering. I think some of us are going to be in person for the first time in a while. So I certainly hope to see you there. I'll be there. And then we will see you again in August. So thanks again for coming, everyone. And we have recorded this call and we will be sending out a link for that in the next couple of weeks. Thanks again for coming.